Okay, we are live as well on YouTube. I'm going to start letting participants in. So
Okay.
Good luck to you both. Good luck to all of us there.
Thank you. Thank you.
Cheers. Natika, you want to share the PPT?
Good morning, everyone.
Good morning, Priyanshi. Good morning, everyone. How are you guys today?
So good.
That's good to hear. So Priya, Rachna ma'am, make sure you guys are active on the chat box and on YouTube as well, if you have YouTube open.
Megha's logged in through YouTube.
She's okay. That's good.
Yeah. So
And then
she
somebody-
asked on the group. Yeah. I told her that she can log in via YouTube and keep getting the questions.
Okay. And maybe one of them also.
And
be
even the
active
maths
in the
team. So maths team also, she asked me just now, Ruchika and all. So I redirected them that if they can be on the YouTube, will help us to coordinate better.
Okay. Priya, I've made you co-host. So if I'm not seeing the participants, like if someone has to be let in, please make sure you are looking at that.
Sure.
Good morning, everyone. We will probably give 5 more minutes for everyone else to join, and then we can get started.
Okay.
Well, let's get started. I think it's about to be 8 past, so we can get started with the participants we have. Thank you guys for joining in time as requested, and everyone else can join us and when they come. So, good morning to you all. Uh, we are super delighted to be kickstarting our direct to student learning transformation series. And today's series is on biology with the title Cell Mania. And we really welcome and we really appreciate that you guys signed up and looking forward to having an interactive and an engaging session with you all. Next slide, Ratika. So a little about Adhyaapan. Adhyaapan is a education organization. We are creating this inclusive ecosystem where in-house educators, industry experts, entrepreneurs all have a unified vision of kindling student curiosity through our what we call the Adhyaapan way of content and learning. What is that? We want to show students why they learn what they learn, and we want to show them real life applications of everything they learn at school. And then through these real life applications, we want to bring to them the career opportunities. And so that all of our content is academically aligned, and we target students from grade 8 to grade 12 from all boards. So the idea is once students learn from a application-based view, understanding where the concepts are being applied, know why they are learning what they are learning, they become fully aware and fully understand the concepts and become innovative. Right? Only if you understand the concepts well, you become innovative. And the second part being, since we are also showing you career choices through these real life applications, hopefully you guys find your passion to one of these career options, and make informed career choices in your life basically. So later, hopefully this you wouldn't be one of those who'd be like, oh, I had no clue why I took engineering, where your passion may have been in something else. So putting all this together, Adhyaapan, our way of learning, our way of content is application-based pedagogy, a career-focused learning, regional language teaching, and an interdisciplinary holistic curriculum. Next slide. So today we have two gold medalist speakers as well, gold medalists at various points in their careers, in their academic careers. We have Ratika with us. She is a Adhyaapan science educator and academic expert in-house. And Ratika, welcome to, and first of all, thank you so much for being with us and putting all of this together. And a little bit about Ratika. So she is a certified science educator and a content developer who promotes experiential learning. Ratika has a undergraduate degree in biosciences from University of Delhi, where she was a gold medalist undergraduate. And then she went on to do her Masters of Science in biosciences as well from University of Nottingham. After this, because of her desire to teach and make sure the kind of learning that student gets can be improved, she came back to India and joined Teach for India and started her teaching journey. Now we are very, very blessed to have Ratika in-house at Adhyaapan. And I'm sure at the end of the session, you guys will know why as well. Welcome, Ratika.
Thank you, Arjun, for such a warm welcome.
Absolutely. And our industry expert for today is Dr. Rashmi Minocha. Rashmi has been another asset to Adhyaapan, and you guys will see why as well right after the session. Dr. Rashmi has her bachelor's degree in biotechnology from DCR University and a master's degree in biochemical engineering from National Chemical Laboratories. After this, she went to Germany to proceed with her PhD in biochemistry where she has done amazing research and won gold medals for her research in Rashmi, correct me if I'm wrong, but that was in gene studies, right?
Yes. Gene expression studies, yes.
Gene expression studies. And she'll tell you more about what those are later during this presentation. After her stint at Germany, she came back to India and joined the All India Institute of Medical Sciences, AIMS, Delhi, and where she is doing path-breaking research in gallbladder cancer for in a very simplified terms. And she has won a 3-year research grant from the Ministry of Human Resources Department, and we are also, delighted to have Dr. Rashmi with us today. Dr. Rashmi, thank you so much for joining us today.
Thank you, Arjun, for the amazing introduction.
Sure. So before I give you guys the stage, just as a perspective on how this session is going to go today, guys, this will be a educational session with Ratika explaining the concepts, but also be a panel discussion between Ratika and Dr. Rashmi, and then you will hear Dr. Rashmi's journey as well. So this won't be just a one-person talking session. This will be a very interactive session, and I'm excited for this sessions to start. Over to you guys.
Thank you, Arjun. Hi, everyone. A very good morning to all of you. Before we proceed, can I request everybody on mute in case you're not? Just check. All of you. Before we proceed, so before we start today's session, I really request students and everybody to have just two things in mind. First, please be very, very participative and interactive. How can you do that? You can probably put it on the chat. If I'll ask a question, in case you have a question for that particular slide, you can do so. In case that question might not be linked to that particular time or slide, we might take it towards the end of the session. The second thing is, I request if you can have a piece of sheet or notebook with a pen, so that in something you are interested to look at later, or maybe something that you have learned, you can quickly jot it down. So just two things that I would request everybody to be ready with. So let's begin with our learning objectives. Can I have everybody saying hi over here so that I understand everybody is hearing me and I'm audible enough? 10 seconds before I start with learning objectives. On the chat. It's already started. Great, Aradhya, thank you so much. I feel that I'm heard. Oh, so many highs. I feel like so good now. Okay, so I'm not just speaking to myself. I have all of you listening to me, and that's a great way to start the session, I believe. Great, thank you so much, Nayana, Diva, Vrinda, Unnati, Sumedha, Sia, Savita, Medhavi, Ashwini, Ashi, and everybody else, Ramani, Diva. Thank you so much. So let's start. So the learning objectives of today's sessions would be, why should we learn more about cells? Why do you have to learn about cells? What is so unique about them? We already know cells are building blocks of life. So why they are called as that? A short comparative study on cell versus atom. Then we have, are all cells alike? Do you think all cells look alike? A recap of types of cells. Some of things you've already done in grade 8. So I'll see if you remember that. Then we'll talk about stem cells and their potential. Study of cancer, which would be talked about in a specific growth pattern, which is exponential growth. Career prospects. And the session would be having career pathways. Also, career prospects will not be towards the end. It will be together with the concept. So you will be enjoying that as well. So let's begin. Please be ready now. I'll be showing you two pictures and I'll be giving you probably a hint in case I don't get an answer. And then I will ask you to use the Mentimeter link when I'll say, I request Priya, please put it over there on the chat when I say So take your time. I'll not give you much time, but I'll not give you even less time. So the first picture is you need to guess what it is. And these are real images. So this is one image, picture one. What do you think this is on the chat? And please don't be afraid to write whatever you think this could be. Please do that. Write it on the chat. And there's a picture two also. I'm putting it over here. Yeah, absolutely, Priyanshi, you're right. They are cells. But I would love to know which type of cells. Okay, cells in a heart. Let's see if that is it. Cell. Everybody is nice. Early telling me they are all cells. Wow, Rasika, adipose cells. Okay, Yashwini, fat cells. Okay. Okay. So you don't need my hint over here. So can I have a Mentimeter link over there? Let's see if you know the answer. I think two answers I've already seen, but let's see if we have Priyanshi. That's beautiful. So let's see if you can type it over there on the chat, on the link that I've shared with you. So I could see so many students just writing their answers and submitting so that I can see how many people know the answer. Yeah, they are cells, nerve cells. Okay, great. All of you are correct. They are cells, fat cells. So many people have answered nerve cells. Okay, heart cells. There's so many answers coming over here. So let's see what exactly they are. I've also given a hint. So in case you want to change your answers, you can. Great. I think most of you know about it, and there are no neurons as well, nerve cells, nervous cells. Okay. So instead of nervous cells, there are neurons or nerve cells. So I think great. Neurons, absolutely stunning. So let's move ahead. I think you guys nailed it already. So yes, there are the images of the cells, fat cells, and neuron or nerve cells, and that's exactly how they look like in real time from our body. So that's the hint that I've also given over there, and just to help you, but I don't think you needed the hint. You already nailed it. So yes, these are the fat cells and the neuron that probably we all see in our textbooks, and you might have seen as well. And I could see adipocyte or adipose tissue being used over there. So let's understand, why should we learn about cells? So anyone who could, thank you so much, Ramani. And I really appreciate everybody sharing their answers over there on the chat so quickly. Why should we learn about cells? Why do we have to know about cells? So as we are all aware, all living organisms are made up of cells or cells. And as humans, we know that they are nothing but the basic building block of life, and they are structural and functional unit of life. Now, when I say structural functionality of life, how do you justify them? Beautiful, Diva. Exactly. To explore more about ourselves, if we know about cells, which is the smallest unit of life in all of us. Yes, Drishti, they are structural unit of life. But now my point is, why are they called as functional and structural unit of life? What do they have? What do they give it to you? Like, why structural? Why functional? Why these two keywords? We always say this out aloud a lot many times, and you have done this in the chapter number 8, cell structure and function in grade 8, if I'm not wrong. Priyanshi, you can put it on the chat. I can see it from there. Absolutely, Ashwini, beautiful. Cell gives structure to our body because they have a specific shape and size, and all of them together, they make something. So they make many cells together, they make something. Absolutely, they perform many functions as well. Absolutely, Priyanshi, beautiful. That's exactly what I was looking for. They make tissues, and then many tissues together which perform same function make organ, and then all those organs finally contribute in the functioning of our body. That is a body. I love the way that all of you are interactive. Lovely to see most of you doing that. So there are three main roles of a cell. Yes, Vrinda, absolutely, they're fundamental unit of life. So they do generate energy. All of us know that. They also make proteins. I think you must have done that in your chapter when you did chapter number 8. And they also make more cells. They also make many cells of like the same kind. They multiply themselves as well. So these three are general and simple functions of every cell that every cell would be doing. So let's understand, why is a cell called a building block of life? I think we discussed it beautifully there. I'm just wanting to show you the cell that you would have seen, and over here you can see that there are many labelings there. So before we come to the labelings, cell must perform basic functions. We just talk about 3 in the earlier slide. So to generate energy that needs nutrients, they also excrete waste, whatever is not needed by the cell. They also respond to the environment, how the environment is around. They also move, they also breathe, they also grow, and they also reproduce. So reproduce meaning they also multiply. So can anyone in the chat tell me all of these: mitochondria, endoplasmic reticulum, nucleus, Golgi complex, all of these are called something. What are they called as? Parts of cells, right? Beautiful, Medhavi, but can you give the spelling correctly? Yashwini, Diva, Drishti, Medhavi, Someda, beautiful. Organelles, absolutely correct. They're nothing but the organelles in a cell, and because of these organelles, so many organelles in this cell, the cell is considered to be a mini organism already, because you have small compartments, small organelles doing some bits that they specialize in, and they are made up of macromolecules. So macro meaning big, and molecules meaning they are a unit that makes the organelles. So they have macromolecules, which helps the organelles to function properly. Prachi, beautiful. Unnati, Sultana, amazing. So we already know about all of this. So if you know everything about why the cell is called a building block of life, now let's connect something that you already know about. Just a quick revision. Nucleus is the genetic material, the place where genetic material or the DNA is stored in. A typical animal cell will have many types of organelles. Can you please quickly put it on the chat? What all organelles do you remember? And probably one of the functions as well. I give 10 seconds. Nucleus is one. What else? Beautiful. Golgi bodies. Mitochondria produces energy. Ashi, lovely. Yashwini, if you could have given a probably a specific function of that. Beautiful, Diva. You're nailing already. You already know things that I have it on the slide. That's so nice. How do you know all of this? Endoplasmic reticulum is the place where packaging and transport facility of the proteins that are manufactured, they are moved or moved inside the endoplasmic reticulum for the entire cell. Yes, Ashi, vacuole, and they are very specific. They are not in animal cells. The question was in animal cells. So we would have vacuole in a cell, but they will be very specialized one. It won't be an animal cell. Cell membrane, Yashwini, beautiful. Lysosomes came, Golgi apparatus came, and there are also ribosomes. I hope you know the smooth endoplasmic reticulum and the rough endoplasmic reticulum. So rough endoplasmic reticulum has those ribosomes into that. Beautiful, Ashi, cytoplasm. You're nailing everything that I really have planned for the session. That's amazing. So all of these organelles are anchored in the cell. So they can be anywhere, right? They need anchoring. They need something to support. So they have a cytoskeleton inside the cell, and they also have cytoplasm that helps them to float in the cell. And all of these organelles are then bound by a membrane that someone also wrote over there in cell membrane. Yashwini wrote, or plasma membrane that supports that outer line for the cell. Lovely. Let's now code what are the type of cells that you know already of. How many are they, and what are their names? You have just 10 seconds. Let's see. You're already so great. You're doing so well. Let's see. Beautiful, Ashi. You nailed one. What else? You beautiful. Eukaryotic. Diva, that's an example of one of the specific type of cells. What does it come under? Muscle cells, yes, but all of these cells will be clubbed into one category. Beautiful. So there are two types of cells. Absolutely nailed it. So there are some examples over here that you probably can use help for. So these comes under the two categories: eukaryotic cells and prokaryotic cells. So eukaryotic, as the word suggests, eu meaning true, prokaryotic, meaning true, and karyotic meaning nucleus. So these cells have a true nucleus. That means they have a well-defined nucleus, and they have DNA inside of it stored properly, apart from other features that these eukaryotic cells have. They're much more advanced than prokaryotic cells. And prokaryotic cells are the cells which do not have true nucleus. Their DNA is, or their genetic material is spread inside the cell. So you can see over here, this is the bacteria cell, which is a prokaryotic example, cell of a prokaryotic cell. And eukaryotic cells, as many of you also wrote about types, animal and plant cells, both of them come under eukaryotic. So vacuole, that one of you wrote, will be a distinction feature organelle for plant cell, which is a eukaryotic cell. So knowing all of this, what could be the possible careers that you can have? That's interesting. So you can become cell biologist because you are so fond of understanding about cell, what happens in the cell, how are these organelles functioning. So you become cell biologist. You can also become molecular biologist, in which you study the cells in their molecular level, like the macromolecules in their molecular level, and understand how are they different in diseased person and normal person. You can become a microbiologist, micro meaning small organisms like bacteria. So you study those organisms and study how they are like causing a disease, and how can you probably treat a diseased patient better. You can also become medical technologist. So medical technologists, what that they do is they take bodily fluids and they test if you have a disease. By testing those bodily fluids, they'll be using many lab equipments to do that. So I've told you 4 professions, 4 career choices already. If you know all of these, if this isn't what interests you. So let's just quickly see this question. Do all cells perform similar function? Yes or a no? Do you think all cells, Priyanshi, that was so quick. No, no, no. Can anyone give me any example on the chat? Like 2 cells who do not perform same function? We talked about cell, and all cells might have, most of them, nerve cell, perform different function. Beautiful. And do they have a same? Okay, spinal cell. So, okay, muscle cell. Great. You already nailed it. So you can see this is the nerve cell or a neuron. Beautiful, Priyanshi. Types of muscle cells I've shown you here, even here. Different functions. Beautiful. That's exactly what is the intent. So even if they have many organelles which might be same, but they still are different in structure and also function. So this is one example that is just a quick revisit for all of you to know about it. So let's make it even more interesting now. Let's connect the cells with something that you might have studied in chapter number 4 in grade 8. Let's see if you can decode it. You only have 5 seconds. What exactly is the picture showing? 5 seconds. I'm making it wow. Yashwini, Drishti, Ashi, Diva, Priyanshi, Drishti, Unnati, Aradhya. That is super cool. That is an atom. Now it can get tricky. Please understand before you answer the question. I'm showing something from this particular picture on the second side. So if you have your finger and you see very closely, you can see very fine lines, which is shown over here. Just follow my arrow there. So you can see very fine lines. Only that you can see. But if I tell you that you might have some superpowers of even going inside, you might see something like this inside these fine lines. What do you think these are shown as? What do you think this could be? Drashti, that is absolutely correct. That is a sodium atom. Cells, so many cells there. So they are so many cells, so they probably are a tissue because there are so many together. Now, if I zoom in, what exactly would be the right side picture? Cells. Beautiful. So they would be the ones which I've zoomed in. So we're trying to now understand atom versus cell. So what are the similarities? What are the differences? What are about the sizes? Is cell smaller? Is atom smaller? How are they different? And we already know cell is the building block of life. What is an atom? Before we move forward, what is an atom? It is also a building block of something. Let's see if you can decode it. I can give you 5 seconds. Beautiful, Ashi. Oh my God. That is superb. I love the way the class is. Like, I'm actually feeling that it is a class-like environment because all of you are so engaged and hearing me out. It's absolutely correct. Smallest particle of a matter. And they are building blocks of any particular thing, which is called a matter. And what is matter? I'm sorry, I'm making it even more simpler because I want to know if your understanding is, it is non-living thing, but it also could be living thing. Is that true? Let's find out probably towards the next slide. Yeah, anything that has mass, or it is a thing, okay, and occupies space. Beautiful, Priyanshi and Yashwini. So matter is nothing but which occupies space, something which occupies space and has mass. So right now, many people actually think that atoms are only part of a non-living thing. So let's understand if it is a part of living thing as well. Let's see. So this is the image that has been taken from one of the research papers, and I would want you to see over here how atoms could be a part of our cells as well. So just see 20 millimeter is the range or is the measurement that you can see. So can you see your finger? Yes or no? Quickly, just tell me on the chat. You can see. So 20 millimeters, beautiful, Priyanshi. And anybody else? I have so many participants. Why only one person, two people, three people I'm seeing? What about the rest? Prachi, that's great. Diva, Ashi, lovely. So you can see your finger. You can see the fine lines as well. So 2 millimeter also you can see. But if I tell you to see all of these cells, would you be able to see it? Rashti, Priyanshi, that is great. Priyanshi, that's great. So, Savita, Unnati, beautiful. You can't see it. So that means from this range of 0.2 millimeter, you're not able to see it through a naked eye. You can't, you have to use some tool to help dig in even further to get that image. So from 0.2 millimeter, you can see that I'm going in deeper to become to a cell. Can you guess which organelle is this? Now follow my laser tip. Which cell, which organelle is this of a cell? Let's see if you can remember from your Diva. Wow. I mean, sorry. I was just thinking that you might not be able to guess it, but you did it. It's mitochondria. Nice, lovely. So in mitochondria, if I zoom in again, I'm now trying to show you some macromolecules that help to put it in function. Prachi, Aradhya, Priyanshi, Ashi, lovely. All of these macromolecules now are making the mitochondria function. They have some smaller units of molecules that are performing a function to make the mitochondria function what it does, like generation of energy. Now, if I furthermore go into this, which is that particular macromolecule, one molecule that I've probably taken, what I see is, what do you think I see over here? Which is a 2 nanometer unit measurement. So imagine mitochondria is 2 micrometer, so you can see 2 micrometer over there, and then 2 nanometer in the unit. Beautiful, Ashi. It is atom. So you can see it's a chemical unit. You can see that I started with finger, and then I moved to the tissue, then I moved to the cell, which is again till cell, it is living, right? And then I'm moving towards the atom also, that right now many of you thought that it is only a part of only non-living things. So it is a chemical, absolutely the aggregation of atoms. And when I zoom in again, the last one, which is 0.2 nanometer unit, it shows me atoms. Now, if that is very clear, let's see the slide which talks about something that you might not know. And how do you appreciate the cell size? So you must have done exponential power in your math class about exponents, and how, you know, exponents talk about something which is bigger in size, something which is smaller in size. So for an atom, you can all see a human cell. You just saw a human cell there, right? So one human cell, imagine it has 1 trillion atom. And how much trillion is? It's 10 to the power 12. That many atoms, imagine the number, it's so huge. And 12 meaning the number is so big. That many atoms are there only in one human cell. And that cell was so small. You just saw over there the range of the cell that we saw. Beautiful. I am actually seeing and sensing the feelings that most of you are coming up with. That's lovely to see that. And imagine a speck of dust. You must have seen a dust sometimes when you pull your curtains down. There's a very fine line of the sunlight falling in your house or in your room. It looks beautiful, and it has smaller particles that you sometimes see. So that is what I'm talking about, speck of dust. It has 3 into 10 to the power 12 trillion atoms. Imagine 3 times more than probably your human cell might have. And the third thing that you probably can think of, yeah, Mehak, how smaller atoms. And you know what? I will try to make you even think how much size can you see an atom to actually visualize and appreciate how small it is. Yes, Diva, beautiful, absolutely right. And the last one is, if you probably have salt and it takes 500 years to, it'll be taking 500 years to just count the number of atoms that probably atoms in a grain of salt would be there. So it will take 500 years for you to count how many atoms would be there in that particular grain of salt. So that's how fascinating it is. So let's watch a video, a short video of understanding how can you connect that atom and the size to your fist. So let's get started.
Atoms are ridiculous and unbelievably small. A single human hair is about as thick as 500,000 carbon atoms stacked over each other. Look at your fist. It contains trillions and trillions of atoms. If one atom in it were about as big as a marble, how big would your fist be? Well, about the size of Earth.
So can you imagine? The video talked about that if the marble is the size, of the atom. So to accommodate that size of an atom, which you probably could have seen, a marble size, right? Small one. If you want to hold that in your fist, and how many atoms would be needed to hold that particular size of the atom? It will be the size of the Earth. So imagine how small the atom is and how big could be a particular object in comparison to the atom that you talked about. Are you clear with that? Like the fist example and the example of the marble? Maybe a yes or a no would work. Like a very simple example I have taken over there. Thank you, Madhavi. Lovely, Vani. Thank you so much. That really helps. Diva, thank you. Priyanshi, all right. So let's move to the next slide.
Atoms are ridiculous.
Sorry.
And
yeah. So let's understand different sizes of cell types. We just saw over there that there are many different sizes that our naked eye can see and the naked eye cannot see. So we just saw over there that up till the range of millimeter to some bit, a little bit more, a bit down, we can see from the naked eye. But when it comes from 100 micrometer to down, you can see that the range is variant. And we also can see the instrument that we can use. So there's one which is called light microscope. Through that, we can see that particular cell, plant cell, animal cell, we can see it. Bacterium, we can see it, even that is smaller. You can see over here, 1 micrometer. So micrometer is 10 to the power -6. When you have minus over there in the exponent, I hope you would have done that. It means it is smaller. And nanometer is 10 to the power -9, and angstrom is 10 to the power -10. So imagine the range is keeping on decreasing. So what is, according to this, what is the smallest bit that you can see under the electron microscope, if I may ask? Type it in 10 seconds. What is the smallest thing that we can see using electron microscope? Lovely. Atom, and 1 angstrom is the measurement, Vani. So 1 angstrom is the measurement, and that is how small you can see through electron microscope. So imagine the power of electron microscope, the power and the focus of the electron microscope that it can use. Absolutely correct. Great. So lovely. All of you are correct. Priyanshi, Prachi, Unnati, Ashi. So by knowing all of this about atom, how can it connect with the cell, what are the possible career options that you can have? One, you can use scanning electron microscopy, basically microscopy, to be analyst. So you can analyze different cells, different sources of the cells of animals or as well as of other living organisms, and you can analyze them for any disease, or you can study them and give the reports as necessary. You can also be a nanotechnologist. So nano means that you are only studying those cells that come into the nano range. For example, you can see over here in nanometer virus ribosome. So only those specific things is what you will study and make life of every individual staying on this earth better. So you can become cytologist. It means studying of cell, specialist in the cell. You can also be in the sales department in explaining to the buyers about which microscope they should get and for what purpose does it serve. So that's why it's microscopy sales specialist. And you can also be a cytopathologist. It means that by studying the cell, you are also telling what disease any individual is suffering from. So you can also become a cytopathologist. So once that is done, can I now tell and ask, probably not tell right now, is atom living? You have 5 seconds. Is atom living? Beautiful, Priyanshi. Of course. Okay. All right. All right, Drishti, Aradhya. All right. Is atom living? Now I will try to help you understand, is atom living or not? After this slide, I will ask you this question again. So let's understand the difference between atom and cell. Quickly tell me, what is the first difference that we talked about? What is an atom? Yes, Drishti? We occupy space and have mass. Beautiful, but I'm talking about atom. I'm not talking about us. Yeah, this is the smallest unit of matter. Is it a chemical unit? It is a chemical unit. What is cell? And what could be the cell? We are living. So what it could be? Yes, it is the simplest unit. It is, the atom is non-living. Okay, Mehak, Mehak, thank you. We'll actually decode it if it is living or non-living. So I've got 2 answers. Yeah, Vasudha, thank you. So cell is biological unit. It's a building block of life, but it's biological. Biological meaning it is living. It has life. And how do you explain that? We'll probably see it over here. So Is atom smaller or cell smaller? We just saw it in the previous one. Is atom smaller or atom? Beautiful. So atom is the smallest bit and cell is larger than that. So we just talked about cells. Cells are bigger. Atoms are made up of electrons, protons, and neutrons. I hope that all of you know about subatomic particles and also many theories of Thomson, Rutherford model that you've already done in Structure of Atoms, chapter 4 in grade 8. Lovely. All of you are saying yes. And Mahima, thank you for giving the thumbs up. So cells are made up of, we talked about macromolecules like lipids, carbohydrates, nucleic acid. Now, atoms are not alive. Why is that? They do not have life. Why is that? Do you think atoms consume water and food to survive? No. So how can they be alive? So the question of are atoms alive or not? The answer is the atoms are not alive. They do not require all of these things to survive, and they are not alive. So that means they are non-living. So I hope that this probably surprises you and your thoughts. So what about cells? Do you think they are living? Justify with the same statement. Yes. Beautiful. Yes. So yes, Mahima, they make bonds. They do not get food and water. They do not move. Even if they do move, the movement is not because of some organelles that they probably have. They are not living. Yes, correct. So they are living. Absolutely correct, Mahek, Yashwini. They are living. They need food and water to stay alive. And because they are a biological unit, they are alive. They have life. That's why you have life. Because cells, because they are living, they make tissues which are living. They make organs that are living, and they make you and the other living organisms who are living. And the next point that makes even further this point concrete is atoms cannot reproduce. Atoms cannot make their own copies. They only can react. As one of them also said, bonding. So they are not alive. Whereas cells can multiply. We know that. Beautiful Drishti, cells can divide and move into two daughter cells. Beautiful. They can form two daughter cells. Now, how many of you are ready for a quiz? Let's see if you know about it. Yes, Vani, correct. Yes, Mahima, through meiosis and mitosis, these are two techniques. So let's get started and understand how many of you, yes, Unnati says, have learned so much. So there'll be a quick quiz. Can I request Priya to please show and share the link? Everyone please join in, and this will be a very quick quiz of probably 3 to 4 questions. Let's see if you can type it in the, if you can answer these questions. The link is there on the chat. Thank you, Priya, for doing that. Please join through this link. Click on this link and you can be directed there. There'll be a name that will be asked. Please type your name and then sign in. Okay. Are you able to log in? Put it a yes or a no there. Just use the link that is given and sign in. Okay. If you're able to sign in, 1 second. Great. So let's start. The first question on your screen. Cell is the smallest chemical unit. True or false? Let's see if you can probably remember this. And time is up. So the leaderboard says, Zanab, you're leading. No, Diva, you're leading. Medhavi, Ashi, beautiful. Next question. Which of the following is incorrect about the function of the cell? Quick. Incorrect. The question is incorrect. Please read the question properly before you give the answer. 13, 12, 11, 10, 9, 5 seconds. Now. Okay, so the leaderboard does say something. Diva, you are maintaining the strike as being second. Medhavi, great. So let's move to the next question. And the question, the answer is it cannot grow is wrong. It can grow. Beautiful. So the ones who have chosen the wrong one, you might not have seen the question properly. So let's go to the next question. There's still more chance to improve. Quick. Go. The question is, cells are bigger than atom. True or false? Ten seconds remaining. Three, 2, 1, and go. So the leaderboard says Medhavi, you are leading. Ashi, lovely. Diva, great. So the answer is true. Cells are bigger than atom. Atom is small. So the ones who have chosen false, please note the correction. So next question, quickly on your screen now. All cells look similar and perform same function. True or false? Quick. I think this everybody should write the correct answer. I don't think there should be a wrong answer. Quickly, 10 seconds. 5 seconds. 4, 3, 2, 1, and go. So the leaderboard says that we have Medhavi maintaining the strike, Ashi on second, and Diva Sharma third. Beautiful. The answer is false. Only one person said it's true. Please revise again. Lovely to see 17 participants doing that correct. So let's continue with the bit over here. So we also talked about many cells, right? So there are some specific cells that we have in our body. So let's see if you can probably decode it from 1 second. So please decode it on the chat. Dash cells are special human cells that are able to develop into many different cell types. This can range from muscle cells to brain cells. In some cases, they can also fix damaged tissues. Beautiful, Vani. They are exactly, Medhavi, correct. They are stem cells. So there are types of stem cells that are there. Anyone who knows what are the type of stem cells that you probably should be knowing and how many they are? I give you 5 seconds. Lovely, Mehak, Yashwini, Ashi, Yash. Lovely to see Yash. You also know about stem cells. That's great to know. Do you know types of stem cells? So there are 2 types of stem cells. One, embryonic stem cells. Yes, Rishti, absolutely. There are 2 types. And the other are called as adult stem cells. So embryonic means that when you're embryo. And how does that happen? Basically, there's a small cord that connects you when you were a baby inside your mom, and that connects the baby through a mom. So that umbilical cord has a lot of embryonic stem cells that can be used to actually cure diseases. And we have adult stem cell, Priyanshi, yes, like in bone marrow. So you can see in this picture clearly that embryonic stem cells are the ones when you are in embryo stage, when you are probably in the development stage. So that time, these cells, which are stem cells, can become anything. They can become muscle cell, they can become nerve cell. That's why they're called as pluripotent. You can see on the red side, pluri meaning many fates. They can be anything. Like just right now, all of you can become anything. You can become engineers, you can become doctors, anything. But when you are having adult blood stem cells, so for example, in bone marrow, there are also adult stem cells. So if I take adult blood stem cells, they can only become something that is related to blood. So that's why they are called as multipotent um, cells. That means they can become something, but only specific to that cell type, which is blood in that case. So they can become platelet, they can become RBC, they can become white blood cell. Yes, Lakshmi, correct. Embryonic stem cells and bone marrow cells. That's correct. So let's watch what are stem cells and why they are important to know through a short video. Everyone, stay tuned.
Imagine two people are listening to music. What are the odds that they are listening to the exact same playlist? Probably pretty low. After all, everyone has very different tastes in music. Now, what are the odds that your body will need the exact same medical care and treatment as another person's body? Even lower. As we go through our lives, each of us will have very different needs for our own health care. Scientists and doctors are constantly researching ways to make medicine more personalized. One way they are doing this is by researching stem cells. Stem cells are cells that are undifferentiated, meaning they do not have a specific job or function. While skin cells protect your body, muscle cells contract, and nerve cells send signals, stem cells do not have any specific structures or functions. Stem cells do have the potential to become all other kinds of cells in your body. Your body uses stem cells to replace worn-out cells when they die. For example, you completely replace the lining of your intestines every 4 days. Stem cells beneath the lining of your intestines replace these cells as they wear out. Scientists hope that stem cells could be used to create a very special kind of personalized medicine in which we could replace your own body parts with, well, your own body parts. Stem cell researchers are working hard to find ways in which to use stem cells to create new tissue to replace the parts of organs that are damaged by injury or disease.
So I hope that all of you now get an understanding that stem cells are undifferentiated cells that have not been assigned a specific function, and because of that they can become any type of cell that you desire, and that's why they hold a lot of potential to cure diseases or to also be able to cure a damaged part or damaged tissue as well. So we'll talk about this more with Dr. Rashmi, as we already have with us. So let's move to the next one. Next slide. So we talked about in the video that they can help you to cure cancer, paralysis, and autoimmune diseases. That's a lovely question, Mehak. So the size of stem cells are also not something which is different from the normal cells. It's the function that the stem cells are and the way they look like. That's how probably we differentiate. And probably that question I also would like to ask Dr. Rashmi about that. Yes, Vaishnavi, you are absolutely correct in both the things. So we will talk about cancer and autoimmune disease moving forward. But before that, we also would love to know about what are the possible careers if we are interested in this particular field. So you can become a stem cell therapist. That means that you can probably suggest that this is the stem cell that you can use to cure this particular disease, or you can also study different lineages of stem cells as well as a researcher. And also you can help with by being a biomedical engineer to help link the biology the understanding with the techniques or the technology together. You can become a researcher, as I just said, and you can also become a biochemist, as we already have with her, with all of us, Dr. Rashmi, who's a biochemist herself. So let's understand one of the diseases, which is rheumatoid arthritis. This is an example of an autoimmune disorder. Auto meaning self, immune meaning wherein the body cells, which are own body cells. Yeah, Mehak, this is a good question. I'll park this question and we'll ask our expert. Right? So we will talk about that session. So I'll keep this question till that point, and maybe we can have that question answered there. So rheumatoid arthritis is one of the autoimmune disorders in which your own body cells will kill your own cells. Absolutely, Mahima. Over here, because the self cells, the own body cells are eating the other or attacking the own cells of the joint. This happens specifically at one point, which are the joints of our body. And why should we know about arthritis? Do you know that 1% of the population of India suffers from this type of disease? So that is almost around 1.8 crore people who have this disease. So we should know about many diseases, but this is a disease which is very commonly found in India, and it used to be related that it is a age-old disorder. That if you are elderly, if one person is elderly, they might have this. There are high chances that they might have this disease. But now the new research shows that it's not limited to only age. Even at the age of 45 you can get this disease because of some professions that you probably are doing. For example, um, a teacher who always is using their joint either to write or to move constantly or carrying many things, so they are using the joints more often. So usage of that and other unknown factors that can trigger a person to have this disease is also established. So male versus female. So females, it's seen that females have more tendency to get this disease compared to male. Yes, Vaishnavi, it's absolutely correct. And that's why it also influences everyday lives. And you're absolutely correct and limits people to their normal functioning. And that's one of the ways that we should be knowing about this disease. And some possible careers, in case you're interested to know about this and maybe do something about it, you can become a rheumatologist, which is a doctor who specifically specializes in treatment of this kind of arthritis. You can also become pathologist. It means you can cure disease by studying about them. So what is cancer? This is the second thing that we saw in the slide where stem cells we talked about. Maybe they can hold a potential. So cancer cells is basically normal. You have many cells in your body, and in your normal cell life, the cells will grow, the cells will reproduce, and when they will get old, they will die usually. But that does not happen when the cancer starts. Basically, even if they get old or even if they get worn out, they will still be multiplying abnormally. So that's where the cancer cells come over, and because of that they become uncontrolled. The growth of these cells become uncontrolled, and that's why we term them as cancer. And because of that, it becomes very hard for our body to work in the way it should. Yes, Priyanshi, and to understand this more closely, we will just see over here quickly about a normal cell and a cancer cell graph. So this graph, the left side that you can see is the normal mammalian cell growth, like mammals. We are all mammals, right? So this is a graph which shows about a cell which slowly starts to grow and then multiplies, and then finally when it gets old, dies. You can see the graph showing that. But you can see in the second graph, where in the cancer cell, can you see the growth? Is it able to die? Please put it on the chat. Do you think the graph shows that the cell death is visible? Death meaning, put it on the chat. Do you see that? Absolutely, Vaishnavi, you're right. No, because the line over here is just going on continuously up, they do not die, and that's why they multiply so much, and that is uncontrolled. Mike, absolutely correct. So that's what is called as exponential cancer cell growth, that they are growing exponentially without a stop. Now let's revisit some of the new terms that we have learned in this session. We have talked about autoimmune diseases. You already knew eukaryotic and prokaryotic cells. We just revisited. You also knew organelles. We also got to know about units of measurement: nanometer, angstrom, micrometer. We also saw types of stem cells, and we also defined stem cells as undifferentiated cells. We talked about how cancer is exponential cell growth. We talked about a little about cancer, and also we talked about one of the autoimmune diseases, which is rheumatoid arthritis. So with this, let's now have a very interesting session with Dr. Rashmi. Can I request now Dr. Rashmi to please join the session, and let's understand more about cancer, and let's start. So please welcome Dr. Rashmi now. We'll have a panel discussion. We'll talk many questions. I'll ask many questions that we all might have, and we can have Dr. Rashmi answering them. Welcome, Dr. Rashmi.
Thank you, Ritika.
So you work in a hospital, right? And you are doing research in cancer.
Mm-hmm.
What are some of the common symptoms observed in patients suffering from cancer?
So Ritika, actually with cancer, there is not a specific set of symptoms. The symptoms of cancer are really non-specific. But if you would ask me to line some of these symptoms, I would say that if you see or feel a presence of a specific lump or in your body or abnormal growth of tissue, then it should get checked immediately. And sometimes there are symptoms like unexplained weight loss or unexplained pain in the body, which does not respond to any kind of treatment, or unexplained fatigue, or any things which are not being able to be treated with normal medication. They should be get checked immediately.
Thank you for sharing that. I also would like to ask, is there any specific cancer case that you would like to share with us as a case study?
Yes, as a case study, I would like to tell about a girl who just got diagnosed with cancer. She had symptoms like just headache. So one day she gets up in the morning, she has headache, and the doctors assume that probably she has some kind of migraine. But when the headache doesn't go after some weeks, then her blood sample is taken and then analyzed under the microscope, and then what pathologists saw that she has a lot more population of blood cells in her blood, white blood cells specifically, as compared to the normal human being. And this is like 20 times more than a normal human being. And then it was diagnosed that she has blood cancer. And then when somebody has cancer, then a panel of doctors will sit and then they will decide what is the specific course of treatment for a particular person. For example, for this girl, there was a combination of radiotherapy or chemotherapy, and then some specific medications for the for generating the immune response.
Thank you for sharing that case with us, Dr. Rashmi. So I would like to ask, and I think many students would have this question in their head, what is the difference between a normal dividing cell and a cancer cell?
So as you can see in the slide, that a normal cell divides quite symmetrically, and while on the other hand, the cancer cell, when it doubles, the shape and size and symmetry of all the cells is quite disturbed here. So basically, the cancer cells are arranged in quite disorganized fashion. This is one thing which you can see and analyze. And then the other main difference is that the cancer cells divide at an uncontrollable rate. So in one particular space, the amount of cancer cells, the number of cancer cells will be even thousands of times more than what body or what tissue can accommodate in a normal person.
Right. So knowing this, I also want to ask, and I think many students would be really wanting to know, how do cancer cells look like under the microscope?
So cancer cells under the microscope look like they're more, as I said, disorganized. So as you can see in this picture, so it's a picture of a prostate cancer showing the cells under the microscope, which are stained. So the blue dots which you're seeing are basically the nucleus of the cells. And on the right hand side, you see a structure which is a normal tissue structure of the healthy person. The cancer has started originating in the left hand side, if you see, and just in the left hand side, the epithelial layer you see, the cancer has started originating, and then you see that the cells are growing in an uncontrolled fashion rapidly, and they are growing in a number which is much more than the number of the cells you see in the right hand side.
Right.
So that's
is it
how you
hard to detect? Great. Thank you for sharing that. I think this is a perfect example of how it looks like under the microscope. Is it hard to detect a person suffering from cancer? And does it take much time to detect cancer?
It is difficult to answer that how long it will take for a person to detect cancer, because as I said, that some cancers come with symptoms, like you will see a lump in the body, and then you will see that it is painful, and then it is a symptom which you can just go and show to the doctor, and then there are imaging techniques which can detect it. But on the other hand, if a cancer has originated in the body, in an organ which is like covered by another organ, which is pressed by another tissue and organ, it is like deep inside, then when it starts, you won't be able to detect it immediately. body. But when it only invades other organs or it goes to the bloodstream, then only you will be able to know that the person has cancer. So that's like an advanced stage.
Thank you for sharing that. Next question that probably I would want to ask is, what are some of the causes of cancer? What are the things that really leads to cancer?
So as outlined by you here, there are some causes of cancer mentioned, which are exposure to the radiations, exposure to some physical or environmental factors, so for example, pollution, and there can be other factors like aging and genetics. Genetics means that some of the cancers are transferred from a parent to an offspring through the genetic genes. So because genes are transferred from the parent to the offspring, and these genes, they carry mutations, and then they will be transferred to the offspring.
Okay. And also, I think over here, smoking is one of the things as well
Yes.
that might lead to cancer. Thank you for sharing that. So we've understood causes of the cancer now. Let's understand one more question. Are all tumors cancerous?
No, not all tumors. Not all tumors are not cancer. So basically, a tumor is just a lump of cells. Okay, and then that lump of cells can just be some fat cells. You were growing fat cells in the body, and then so these cells, these tumors which are not cancerous are called benign tumors. And how do we know that a tumor is benign? So it will grow slowly. It won't be having a rapid cell division, and it will be just restricted to one place in the body. So it won't be invading surrounding tissues. It won't spread to the distant parts of the body. While a cancerous tumor will spread quickly, so it will divide very quick, and it will often invade other surrounding tissues of the body. It will spread to the other parts of the body, either through the bloodstream or through the lymph node, and it needs treatment. So if left untreated, the person can die.
Okay, got it. I think that's really very informative about knowing if not all tumors are not cancerous. So what are cancer stem cells?
So cancer stem cells are a population of the cells in the. So if somebody has tumor and I take the tumor out of the body, then you will see that there are some normal cells which are rapidly dividing, and then there are subpopulation of the cells which specifically are able to cause cancer. So they are basically the initiating points of the cancer. So if I take these cancer stem cells out of the tumor and then put it into another organism, for example, a mouse, that mouse will also get cancer. So these are the cells which are basically resistant to treatment. So when we have chemotherapy, when a person gets a radiation therapy, these are the cells which are very much resistant to the treatment. And then even if one cancer cell is remaining after the treatment in the body, then the tumor can reoccur, as you can see in this image.
So you're saying that if one person is cured of cancer, it can happen if the cancer comes again?
The cancer can relapse in the case when all the cancer stem cells at the point of the tumor have not been vanished or have not been taken out or destroyed.
Thank you. There are some questions that the students are asking. I would like to ask: can stress cause cancer?
There are not any specific studies which say that the stress can cause cancer, but I think that stress can cause some changes in your epigenetic changes in your chromosomes, in your DNA, which means that these are changes which are not directly in your DNA, but these are some environmental or changes which come from the environment. And if there is so much of stress, and stress is at that level that it is causing mutations or changes, epigenetic changes, then there may be a possibility that it can cause. So healthy body needs a stress-free lifestyle, in other words.
Thank you so much. I think it answers. Also, one of the questions was also this: is cancer only curable at initial stages?
Cancer, yes. If cancer has not been reached to other body organs, then there is a possibility that we can cure the cancer at the initial stage, but it differs from case to case. So if, for example, cancer is restricted to only one part of the body, we can just take and remove it by the surgery, and then the cancer is gone. And but say if this cells have started invading the nearby tissues, nearby blood vessels, and they've started growing to the other parts of the body, which is called metastasis. So they're invading other organs of the body. Then even if one cell goes to the other organ, then it will start causing cancer to other body organs. And then at that stage, it is very difficult to find a suitable cure.
Beautiful. Maheshwari, I think it answers your question. If it is in fourth stage, the curing bit, I think what Dr. Rashmi is saying is that in case it doesn't spread through the entire body, it can be cured. In case it has, it becomes very difficult to get cured because it has already spread to the entire body. Is that correct, Dr. Rashmi?
Yes.
One
Yes.
thing also, if you can tell about any cancer which is most curable. Which cancer is most curable? Like high success rate.
So I think by seeing that which cancer is most curable, rather than answering this, I can answer that the cancer which has more survival rate. So for example, prostate cancer or thyroid cancer, these are some types of the cancer which has more survival rate. We cannot tell that we can cure the person completely, but we can just improve the survival rates.
Okay. Great. So one question also coming that, so blood cancer is more difficult to cure than lung cancer?
Yes, because when somebody has blood cancer, then because blood is something which is going to every organ of the body, and when blood is going from one part to another part, it can carry the cancer stem cells, and then it can invade other organs in the body, and then that's why.
Beautiful. I think lovely. I think we really understood that. So we just talked about stem cells, and could you elaborate more on how can stem cells be a potential therapy or a source to cure cancer patients?
As you already heard that stem cells have the capability to build every tissue in the body, so which means they have the potential to can be used as therapeutic purposes for regeneration and repair. So stem cell therapy have been already used as in cases of bone, which are also called bone marrow stem cell therapy, in which the patients with the blood cancer can be cured by taking the stem cells, so providing the new stem cells into their body. So either these new stem cells can come from a donor, from identical twin, for example, or the stem cells can come from a person's own body. So for example, if the cancer has not spread to bone marrow or every other part of the body, and the cancer is like to one part, and then we can use patient's own body cells, stem cells, which are still healthy. And then these cells are taken out from the bone marrow, and then they are grown under physiological conditions in vitro lab conditions, where they are separated from the other cells of the body. So we have only stem cells and not like other cells. And then these stem cells can put back into the body. But before you put stem cells back into the patient's body, the thing which should be taken care is that the patient has to undergo first destroy the unhealthy cells. So for example, if a person has cancer, then that person has to go through chemotherapy or radiation therapy, so that the cells which are unhealthy have to be destroyed first. And then when you destroy all the unhealthy cells of the body, then basically you are deprived of, for example, white blood cells, which make the immune system. In that case, then we provide new healthy cells, which are stem cells. Then we will make more blood cells in the body. And then this is something which have been recently used to cure blood cancer patients, although it is like very, very costly treatment.
Great, thank you for sharing that. There are a few questions. How to identify the symptoms of cancer?
Sorry, again?
How to identify symptoms of cancer. So we just talked about, I think you just talked about few things that probably might be the starting point, like fatigue, lot of fatigue.
Yes. So if you have something which is like unexplained by normal tests, you just get a blood test and you get a normal, for example, you are losing weight, and that is unexplained, that you don't, without any exercise, without any changing in the food routine, you are just losing lots of pounds, and then that is something like unexplained. And there can be reasons like unexplained fatigue, so you are having a loss of energy, and you even to that extent that you're not able to do daily activities in your life. And then there is something which, yes, but not, it doesn't mean that every such symptom can lead to cancer.
Correct.
Said that there is very unspecific set of symptoms.
Thank you. So, is skin cancer also difficult to cure like blood cancer?
I think skin. It depends on how at what stage
stage?
person is.
Which
But
stage?
yes. But because yes, but there are instances where there are clinical trials where people are using
Okay.
stem cells to cure.
So, is this treatment of stem cell therapy expensive?
Yes, it is a quite expensive treatment because stem cells are quite difficult to, we have a very small amount of stem cells in the body and they're very difficult to take out and then grow in the artificial conditions. And then the success rates are not like 70 or 90%. The success rates are like 30 to 40%, something like that.
Okay.
And then
So
it's like probably lakhs and lakhs of money you need.
it's expensive. So,
Yeah.
one question over here is, if someone is clean from cancer for a long time but suddenly develops cancer, what might be the reasons from regeneration of cancer after a long time?
So sorry, I didn't get your question again.
If someone is clean from cancer for a long time, but
Mhm,
suddenly the doctors detect
mhm,
cancer again,
mhm,
what
mhm.
might be the reasons for regeneration of cancer after a long time? I think you explained that.
Yes. If even if so sometimes what happens that you give radiation, all the cancer cells there are destroyed, but somehow there is some cancer cells which are still there, some tumor cells which are still there. Either they have gone to the bloodstream or they have gone to another organ and they've started invading it. So, but you won't know it in the beginning. But probably if it's like sometimes it happens that you get cancer, then you get cured, and then after a few years later that you find out that you still had some cells remaining which were not treated at that time, and they've gone to the other organ and then they have invaded other organs. So that's how you can. Yes. And yes, there's of course genetic possibility. So if somebody had cancer, then there are techniques like sequencing techniques. So you can sequence the gene of the person if somebody has having cancer in their generations. Then we do gene sequencing, and then we know specific mutations which are there present, and then we know that that person might have chances to have cancer in
To
his
get
or her
it.
lifetime.
Thank you so much. I think we learned a lot from you from through these questions and from your experience. Thank you for sharing the expertise of about cancer cell with all of us. We will have shortly. We'll be shortly inviting you again to talk more about what you do in just quite a bit. So thank you so much, Dr. Rashmi.
Thank you, Ritika.
So let's recap what we just saw. So cancer is abnormal growth of cells. Every tumor is not cancer. Causes of cancer we talked about. There's no one specific cause of cancer. There can be many, and there can still be some other unknown ways as well through which cancer can happen. So it's not limited to only the ones that we saw, but they are more or less the causes that we usually see leading to cancer. And we also talked about possible treatments. So radiation, using radiation like X-rays. We also talked about chemotherapy. We also talked about stem cells being a potential therapy as well. So possible careers that you probably can have. If you know if this topic interests you, you can become a cancer researcher. You can be a doctor specializing in cancer treatment called as oncologist. You can also become cancer. Sorry, you can also become cancer scientist who studies on cancer. So the learning summary for now, till now, whatever we have learned was about cell. We also talked about how cell and atom are related, some similarities and differences. We also talked about cell types. We also talked about our all cells alike, and you did lovely in the time of the quiz. We also talked about undifferentiated cells, which are nothing but stem cells. Then we talked about, sorry, we talked about potential of stem cells in curing diseases. We also talked about a growth curve of normal cell versus cancer cell having exponential growth curve. And lastly, we also had many career choices, prospects along with all of these learned concepts. So summary of all the career prospects that we talked about were all of these. You can maybe have a quick read. I just brought all of those individual ones on the previous slides to come on one slide so that you can know, just by knowing about cell and what a cell does and the potential it holds in the stem cell and the techniques that are used to study cell, all of these are possibilities that you can probably choose from. It's very diverse. So moving further, I welcome again um, Dr. Rashmi. So as I said, as I promised, we'll now know and understand the life of a biochemist and more about it. Dr. Rashmi, so the first slide is how do we see cells in a lab? I think most of you already know now how do we do that, but can I have Dr. Rashmi speaking about it?
So as Ratika told that we use microscopes to see cells, and usually when we work in the lab, we work on cell culture. So we grow the cells in an artificial environment. And for that, we use mostly optical microscopes, which provide magnification like 100 times more magnification. And then there are more advanced microscopes, which are very, very costly and need specific treatment to cells before we can see them. And there's our electron microscope, which use electron rays, beam of electron to see an image, while an optical microscope just uses light waves. So for all lab purposes, for viewing live cells, we use optical microscope or different types of optical microscopes.
Beautiful. We really would love to see some samples of cells that you probably would be doing in your lab. So can we see some cell samples?
Mm-hmm. So here's a time-lapse light micrographic footage of the nerve cells, neurons, in the microscope. So can you just play the video? So you see that in this video, yes, neurons in the culture. So they're forming a network. So if you see the videos, they are growing. So you see the nerve cells, you see that these are surrounded by dendrites, these extensions, and then the axons. And then so how the nerve cells are basically connected together and how the information is being passed from one cell to another cell. So it's very beautifully here.
Beautiful. I think we just saw that. And these are also some cells that
yes, these are specifically stem cells. So basically here what you see is a difference between a normal breast tissue and a tumor tissue and the surrounding lymph node, which is also invaded by cancer. So you see that in the normal breast tissue, you see that the structure is quite organized, and you see a limited number of cells, and they are very symmetrical. While in the tumor, you see under the microscope that the cells are going indefinitely in all directions and unsymmetrically. So if you further zoom this, because this is not zoom, if you further zoom it, then you will see that the nucleus is also dislocated or disorganized, and they're bigger in size. Sometimes so you can even see the differences between the cells from a normal tissue and the cancer tissue.
Great. So can we know more about how you work in the lab and your ongoing project?
So right now we work in a cancer biology lab. So we work in various types of cancers, and how we do that? So we grow cells in as 2D culture and 3D culture. So it means that we grow cells in a sheet or layer, and then we also try to grow cells to stimulate more 3D kind of environment, like in the body. So which is a recent thing, recent research area. So what is the purpose of growing cells? So we have, for example, surgeries or tumor patients in the hospital, and then these tumors are excised out of the body, and then we can use these tumors for research purpose by extracting the cells out of these tumors, and then we have the single cells. We can grow them in the incubator in special, giving them special media and serum requirements. And when these cells grow, we can use them for first studying the invasion or proliferation characteristics of the cancer cells, how they are invading, how they are proliferating, how they are basically growing and causing cancer. And the second, we can use these cells to find out test some drugs, so run some preclinical trials. So preclinical trials means that we just take the cells out of the body and then supply drug to them, and then we see that whether this drug is working, whether the cancer is shrinking or not, and that there are specific kinds of tests and markers to test that. So here in this image you see that organoid, which is like an organ kind of structure, which is prepared artificially using the cells from a tumor patient or from a cancer patient, and then we grow them in the artificial environment in an incubator, and that's how it looks like. So it looks like at 3 different time points of the 2 different cell lines, and then they will mimic basically the cell-cell interactions and the environment of a tumor inside the body. So it looks like a tumor. It is a tumor, but it is like a very
out of the body.
Yes,
out of the body.
So it is created for a research purpose so that you can test drugs and then find out what kind of drugs will work.
That sounds so fascinating. I think all of us are very thrilled. How can cells be really out, and all of these can be mimicked outside of the cell, out of the body, and cells be still be alive doing that. That's lovely. So can we go to your lab and see what you do in your time day to day ways? So can we just talk about how your lab looks like and what do you do?
So I just took some pictures of lab, and then so I just put it here, and then you can see that the first picture, there's a -80 freezer where we store our samples. So the all cell samples and all, either they are stored in liquid nitrogen or they are stored in the -80 freezer at quite low temperatures. And then there is a laminar hood where we do our cell culture experiments. And then there are some specific experiments going on here with the cells. And then there are some instruments which I just clicked photos of some lab instruments. So this is a microscope which is attached to a computer. So you do not always have to look in the small lenses to look at the images. So what the microscope will do, we just put the specimen and then just connect it to the computer, and then whatever is shown in the microscope you will see in the computer. You can just save images, you can just make a video of the cells also. Then on the left-hand side, you see a nanodrop machine, which is a machine to quantify how much RNA or DNA or protein is present in a particular sample. So you just put a drop of that sample and then it will tell you. And then on the right-hand side, you see a real-time PCR machine. So real-time PCR machine, you have heard about it during the COVID times. It is used for amplifying a specific gene. So polymerase chain reaction in a quantitative terms. Yeah, and it was used for the COVID diagnosis, right? You remember it. So this is how it looks like. So it is also connected to a computer, as you see. Then there is a centrifuge on the left, you see, where you spin down the cells, and then you, for example, you have cells and then you have organelles and everything, and then you want some specific organelle, or if you want the cells to be differentiated from the other parts of the, the cell, other parts of the tissue or something, then you use the centrifuges. And then on the right hand side, you see a sonicator, which is used to shear down the DNA. And break the cells, to lyse the cells, to get the things out of the cells. Yes. So the next slide, please. Then on the left hand side, you see an ultracentrifuge. On the right hand side, you also see another centrifuge. So these are powerful centrifuges, which can rotate at a very high speed, and then they can separate the cells from their constituents. And then this is an advanced level machine, flow cytometer, which is used for cell sorting or cell cycle analysis. Yes. And both are the 2 different types of the flow cytometers.
I think it's so fascinating to see your lab so nicely, and the pictures are so clear that I think it just inspires all of us to be in the lab and start working just now. I'm sure the students would be feeling the same thing. Thank you so much, Dr. Rashmi, to share all of this. So with this, let's charge ourselves up for another quiz, and you can buckle up. Let's see how many of you will really like listening to what we are learning. And thank you, Priya, for sharing the quiz link. So let's start the quiz 2. So please be ready to join in as quickly as you can. We'll start in 5 seconds. 4, 3, 2, 1. Only 4 people have joined. I request everybody to start joining in. 5, 6, last 4 seconds. 7, 8. Okay, I'm starting now. 3, 2, 1, go ahead. Stem cells are, choose it. Please get all the questions correct. Let's see if all of us can do that. Last 5 seconds. 4, 3, 2, 1. Oh, all done. Okay, so the leaderboard says something and we'll have answer as well. Leaderboard says Ashi, great. Okay, Vani is the high scorer. So the answer is undifferentiated cells and can become any cell of the body. That's the correct answer. Many of you have done that, and the other options were confusing for a reason. So please focus on the next question and read the question before you answer. And go ahead. Cancer cells are. Please choose the option correctly and quickly. Quickly, 20 seconds. Quick. There are few more people waiting. Quick, quick, quick. 10 seconds. 2 more people, 3 more people. Now 5 seconds. 4, 3, 2, and time's up. Okay, so let's go ahead. Leaderboard. Vani, you are nailing it. Madhavi, great. Second, and then Aradhya. So next question. The answer was they cannot grow. Second option was the answer. Or tool that can be used to view atoms or electrons. I think this should be correct from everybody's side. Something that is used to view atoms and electrons. Please read the question properly. You have seconds, less than 20 seconds. 10 seconds. Okay. So the leaderboard says, Vani, you're still maintaining the strike. That is so nice. Someone named Lentil has been now second. So electron microscope is the correct answer. Great. Lovely. Only one person. Light microscope, not really. You can't see atoms and electrons from there. Last question now. Over on your screen. 3, 2, 1, and go. Causes of cancer can be. Please remember that slide. And we talked about it a lot, many times. So I hope everybody nails this as correctly as you can. Still have 15 seconds. Quick. Okay. So the answer for first the leaderboard will be talked about, and then we'll be talking about the answer. Vani, you have maintained the strike. Aradhya second, and then you have been now the third one. So the answer is all of the above. Smoking was only one cause, but I think all the 3 were right. So all of the above is the correct answer. Lovely job done. So with this, I just want. I think all of us have a question: how to become like Dr. Rashmi, so inspiring, and what she does is extremely commendable. So I invite you again, Dr. Rashmi, could you please share out what are the possible options after 12th class, and how do we really become something like you?
Ratika, thank you. So in order to become a scientist or a researcher, or there are 2 pathways broadly mentioned here. So if you really are into biology, and then you want to become a doctor, you want to deal directly with the patients, and then you take biology in your 11th and 12th standard, and then you try to give the exams for the MBBS. And if you are really not sure whether being a doctor is what you want, and you are more interested in studying biology rather than treating patients, finding or exploring out the questions which are still unanswered, then you can also take another path, which is like doing undergraduate studies in a life sciences field. For example, doing Bachelor of Engineering, Bachelor of Technology, and Bachelor of Science in a field of biology, like I did. So I did my bachelor's in biotechnology, while there are some other fields like biochemistry, microbiology, zoology, botany, genetics, etc. Then once you are done with your undergraduation, then you can also do a master's in a specialized field of the biology, followed by a PhD. So where you will work on a specific problem or a specific question. So in a PhD, you make a hypothesis about a specific problem. You are working on one single protein, for example, one single disease, one single mutation, or one question you will just ask yourself, which you find really interesting, and then you work for the next couple of years answering that in a technical manner, using the technicalities of the cells and tissues, and working on the real experiments. That's what you do in PhD. And post PhD, so I'm right now in working as a postdoctoral person in the medical institute. So in order to reach that stage, when you're doing PhD, you will know what exactly you're liking or disliking, what further field you would like to continue.
Great. So a quick question: how many hours did you put in when you were as a student, like all of us, and then shaped your career to what you are right now? Like how many hours and years of input that you are already given to become what you are? It will really help us inspire.
So talking about years, I would say that graduation takes like around 5-6 years, and then a PhD depends on what kind of problem you're studying or where you're doing your PhD, but usually it takes like 4 to 5 years on an average. So to become a scientist, I would say it will take 10 years after you pass your 12th, and then it depends on how much experience you have or what you're looking for what kind of job you want to do, work in an industry or you want to go in an academia. Talking about number of hours, I would say that rather than counting hours, you find out how much focused you are on one particular objective of your life, what you want to do, and always try to find a reason why you want to study, not just because you want to pass an exam, but because you really have passion for something. So that's
a wonderful advice, I believe. So what are the institutions that could help us be a biochemist?
On the screen, you see that there are some institutes which are already mentioned here. So these are some of the institutes which are top institutes in India, which are working in the field of scientific research on cells and on related topics. So you see that there are all IITs, NITs, and medical institutes like AIIMS, and then there are specific research institutes like the Indian Institute of Scientific Research or National Center for Cell Sciences, which is working specifically on cells. So there are other institutes also which are not mentioned here, but these are the most popular institutes of India.
Great. So Diva, to your question, this is the last bit that in any bit anyone wants to know about the organizations. This would be, we're just almost to the closure. Diva, I hope it answers the question. So some of the organizations, if you can suggest, we can work after we have completed the education which needed to become a biochemist.
So this slide gives you an idea about what kind of work you can do after you completed your PhD. So either you can work as a scientist in a research institute or in a medical institute in a lab, and work on a specific problem, find out discover a new drug for a disease condition, or you can work for a pharmaceutical company or a biotech company such as Bharat Biotech. They can help in the production of a new vaccine at a large scale level. You can also teach and join a university as a lecturer or professor, or you can also, join companies which provide scientific instrumentation, scientific reagents, or consumables, which are also an important part of the research, because without that, research cannot happen. And there are some government funding agencies which provide funds to a specific individual if you want to carry out your own research, and then you can set your own lab and work on your own topic. You can just submit a proposal to government, and then if the proposal is, if the government likes your proposal, they will give you money for setting up a new lab. So you can set up your own lab.
It's so inspiring to know so many things and what all can we do if we know all of this. I think all of us got a lot of knowledge today to understand just about cell and how important it can be and in making informed choices about our careers. Thank you so much, Dr. Rashmi, for sharing. I request Arjun now to take over. Arjun, over to you.
Sure. Hey, that was an amazing session. Thank you, Ratika, and thank you, Dr. Rashmi, for giving us that. Just the sheer interactions between the students, all the questions that the students were asking. I'm sure there are still a few questions that Dr. Rashmi and Ratika yourself can answer, and we can take that up in a few. So thank you guys, for all this engagement so far. Q&A will be coming up, but before that, I would like to share with you the upcoming sessions from Adhyaapan. We have one session on chemistry, which is the metallurgical magic, coming up next week, and Priya will be sharing the sign-up forms in the chat box. And then right after that, we have the physics session, which is on force and motion, on 24th of April at the same time. So we really hope you guys attend these follow-on sessions as well, and maybe spread the word around for more people to come and join if you thought that the session was informative for you guys. Next slide, Ratika. I think that's it. This was a thank you slide for sure. I think before we say our final thank yous, let's take up some questions from the audience, please.
Sure. There was one question that was asked about stem cell. Dr. Rashmi, if you probably can share. There was a specific question: how do we identify stem cells? Do they have a different structure? And if you probably can share some of the like how small are they? Smaller than the normal cells? How are they different? Like how do you recognize them under lab conditions?
Thank you, Ritika, for the question. So stem cells are, as I said, that stem cells are basically the cells which can differentiate from any body cell. They would be difficult to distinguish like directly under a microscope with a normal cell or cancer cell. Even a cancer stem cell and a stem cell would look alike. But how do we isolate them is that then there are specific bodies, and these stem cells actually on the surface of their cells they have specific proteins which are present only on the surfaces of the stem cells. And then there are antibodies which have been discovered which will just bind to these surface proteins, and then they will just take them out from a population of different cells. So that is a way you take stem cells out of the whole cell population.
Great. I hope it answers to one of the questions that came when I was explaining stem cells. So any other question that our students or anyone would like to share or ask, please put it on the chat.
There was one question from Zainab. I think Dr. Rashmi answered that. There was one question that was asked by Aradhya. How is it possible for a newborn baby to be infected by cancer?
Yeah. Thank you, Arjun.
So it is a very, very rare chance that a newborn baby, if a mother has cancer, that the cancer will spread to a newborn baby, because we have placenta where the baby is growing, and there are not many ways by which the cells can just transfer directly to the. I think there are only very, very few cases in the world. In the whole world, like probably 10 or less than 10, where the newborn babies got cancer from their mothers, but through the placenta. Placenta is the only way, because placenta is the layer which helps to transfer nutrients from the mother to the baby, and it is very uncommon. It is not. There are not so many instances we have seen like that.
Thank God it's uncommon, right? Like, that's the most meanest thing one person could get. I think from the other questions, people, I think Priyanshi was asking about what might be the reasons for regeneration of cancer.
Yeah, we
after
talked
a
about
long
it.
time.
Yeah,
We
we
spoke
talked
about
about
it.
it.
Okay. Any
I
other
think,
questions, anyone? I think Dr. Rashmi had answered pretty much all the questions here.
okay. Yes. Okay, if probably you don't have any question, can I request probably in one word, if you can probably help us understand how was the session, and if Arjun you agree, like we can end the session by knowing in one word how was it, that'd be lovely. I think
Absolutely.
that we can have. Over to all of you. Thank you, Ashi. That's a really good adjective to use. Thank you. Thank you, Vani. Your feedback really helps us to grow and to understand the needs and how can we be better. Thank you, Yashwini. That is really helping us to boost our confidence and how can we be better. So only 3 people, we can probably have more. And lovely interaction in the quiz. I think it was fantastic how all of you were so tuned to the quizzes, and most of you were hearing, and that could be seen in the quiz results as well. So lovely job. I think you guys nailed the 2 norms that I had set. One being participative and the other being if you would like to add anything or note down some of the information. Thank you, Ramani, Fia. Thank you, Sultana. Please take 5 minutes. If you can, just copy the link, the feedback form, and if you can fill it, it will really help us to know the data really quickly. And it is a very simple form. It doesn't have, it's something like a quiz that you just did, lovely and short and crisp. If you can do that, it will be amazing. So we just have shared the link as well.
Yep.
That's a quiz result for all of us. So we hope that you can do that.
Yeah, guys, please make sure you fill out the feedback form. It helps us a lot. We are a budding organization, just started just over a year ago, actually. And so any of your feedback, anything at all will help us grow to make sure that, we offer sessions which are informative and awesome and wonderful, like you guys have mentioned, and more sessions to follow so that these adjectives can keep flowing. One thing that I also want to mention is I will be sending you guys in the WhatsApp group or over the email a subjective assessment. So there will be questions from the session today, and you guys will get that questions. There's no need for you guys to send it back to us. This is just to make sure that you guys have understood the sessions and can answer the questions. And after a few days, we will also be sharing a answer key, so that just to for you guys to know that you've answered the questions correctly, basically. So if that is it, like I am super happy with the way the session has gone. Ratika and Dr. Rashmi, thank you so much for conducting this in a very, very engaging and a highly energetic way. And guys, this is the kickstart for our D2S sessions, more sessions to follow. And I would love to have you guys again, and more of you guys actually, for the upcoming chemistry and physics sessions. And then we will have math sessions to follow as well. So with that, you guys have a great rest of your day. Thank you for joining us. Even though you guys have, most of you guys have puja at home today. So thank you guys for joining us, and you guys have a great rest of your day, and stay safe. Cheers.
Thank you, everyone. Bye-bye.
Bye-bye.
Hi, Fia.